The overall goal of this procedure is to measure mitochondrial function in living yeast cells using two green fluorescent protein, or GFP, variants. Redox-sensing GFP contains surface-exposed cysteines which undergo reversible and environment-dependent oxidation and reduction, altering the excitation spectrum of the protein. Reduced Rho GFP has an excitation maximum at 470 nanometers, whereas oxidized Rho GFP has an increased efficiency of excitation at 365 nanometers. The redox state is measured as Rho GFP emission at 470 nanometers excitation over the Rho GFP emission at 365 nanometers excitation. GoA-Team is a FRET probe in which the epsilon subunit of the F0-F1 ATP synthase is sandwiched between a FRET donor GFP and a FRET acceptor orange fluorescent protein, or OFP. Binding of ATP to the epsilon subunit results in conformation changes in the protein that bring the FRET donor and acceptor in close proximity, allowing for energy transfer from the donor to the acceptor. ATP levels are then measured as the OFP emission upon excitation at 488 nanometers over GFP emission upon excitation at 488 nanometers. Results are obtained that monitor changes in redox state or ATP levels that occur under physiological conditions.
The main advantage of these methods over existing methods is that Rho GFP and GoA-Team are genetically encoded and can be introduced and stably maintained in living cells. As a result, they can be used to measure mitochondrial redox state and ATP in living cells without affecting the fitness of either the cells or the organelles.
Both probes monitor changes in redox state or ATP levels under physiological